In first part of sexual reproduction in flowering plant, we explained the typical part of the flowering plant. And in second part, we discussed with respect to the stamen, anther and microsporangium structure. Whereas the third part, we explained pollen grain structure, pollen grain size, pollen grain definition, then along with that pollen allergy, bee pollen, pollen grain viability, cryopreservation and also microsporogenesis. Then in fourth part, we have to discuss in detail the female reproductive whorl. In that first we have to consider what do we mean by pistil. Then megasporogenesis. Then development of embryo sac. And finally, structure of ovule. Ovule we also called megasporangium. First, we consider the term pistil, that we also called gynoecium. Pistil or gynoecium is a female reproductive whorl or female reproductive part it is. Structurally, it is composed of a carpel as a unit, each carpel having basal ovary, then style and stigma. If you consider typical pistil which possess basal, the cup-shaped ovary, then style, finally stigma. If you consider the stigma, it is a cup-shaped structure and mainly receives pollen grains. Hence, it is a receptacle where it receives the pollen grain of same flower or another flower during pollination. Style, generally long, slender structure. It keeps stigma in particular height. Therefore, it connects to both ovary as well as stigma. Then last part is ovary, which having the specially ovule. Ovule become future seed. At the same time, this ovule lodge in a particular chamber. That chamber is called locule. Then the ovule get the nourishment from a specialized tissue, that is what we call placental tissue, and that structure is called placenta. It is a typical pistil, or in other words gynoecium. In many plants, only one ovary, one style, one stigma is there. That condition is called monocarpellary. Means the monocarpellary pistil only having one ovary, one style, one stigma, or single carpel we say. If it having more than one carpels, that what we call multicarpellary a pistil, it possesses more than one. At that time, there may be a fused, what you say, carpels are free. If carpels are free, at that time it is called apocarpus. Means carpels are not fused, they are free. They may be syncarpus, where the carpels are generally fused there. You take in case of Mycelia, now then Sampige anta idu. Many carpels are there, these carpels are free. At the same time, in case of hibiscus, we also call it as a China rose, Dashwal anta ಹೇಳ್ತಾರೆ, and papaver somniferum, we called poppy plant. ಅಥವಾ ನಾವು ಕಸಕಸಿ ಗಿಡ ಅಂತ ಹೇಳ್ತಾರೆ. In these two examples, generally many carpels are there, they are fused, that condition is called syncarpus. Next, we have to consider megasporogenesis process. In case of ovary wall, a sporogenous tissue is developed. That sporogenous tissue later gives a specialized cell we called megaspore mother cell. That megaspore mother cell we called MMC. This megaspore mother cell undergo meiosis. By first meiosis, two haploid cells are formed, and later by second meiotic division there is a formation of totally 4 haploid cells, and these 4 haploid cells itself we called megaspores or megaspore tetrad, because they are 4 in number, hence we called megaspore tetrad. Hence the process of formation of a megaspores from a megaspore mother cell we called a megasporogenesis. It takes place by meiosis. If you consider these 4 megaspores, they are all haploid in nature. In most of the plants, the one megaspore develops into female gametophyte or embryo sac, and 3 degenerate. That condition generally we call monosporic, we say. Then majority of the plants shows monosporic condition. What happened? Any one of these 4 haploid megaspores develops into a embryo sac, remaining 3 degenerate. Then in some plants there is a development of 2 megaspores into a embryo sac or female gametophyte, and 2 degenerate. That condition we called bisporic. Whereas in few plants 3 megaspores develops into female gametophyte and 1 degenerate. That condition what we call a trisporic. And very rarely all 4 megaspores develops into embryo sac or female gametophyte, and that what we call tetrasporic, and this what is as a concept was explained by a great Indian botanist P. Maheshwari. Panchanan Maheshwari classified the development of female gametophyte or embryo sac into 4 types. That is, one is monosporic. In that condition, one megaspore develops into female gametophyte, 3 degenerate. In bisporic condition, 2 megaspores develops into a female gametophyte, 2 degenerate. Whereas in trisporic condition, 3 megaspores develop into embryo sac, 1 will degenerate. Whereas in tetrasporic condition, all the 4 megaspores develops into 4 different female gametophyte or embryo sac, and that condition is called tetrasporic. Now we have to discuss the term development of female gametophyte, or that we also called embryo sac. As we know, in most of the plants, out of 4 megaspores, only 1 megaspore develops into embryo sac. Hence, that haploid megaspore, which is produced by a process called megasporogenesis, enlarges little bit, and it forms the sac-like structure. Hence, female gametophyte also called the name embryo sac. This haploid, a single shell, first only undergo nuclear division. By first mitosis, there is a formation of 2 haploid nuclei. Then, once again, by second mitosis, there is formation of a 4 haploid nuclei. These 4 haploid nuclei formed by the mitotic division, hence the number remain same. Whereas when third time mitosis is take place, totally 8 nuclei are formed. All these 8 nuclei, which are formed during the female gametophyte development, haploid in nature, or they contain 1 set of chromosomes. Now, after completion of nuclear division, there is a rearrangement of these nuclei, and also a protoplasmic division is take place. Here, out of 8 nuclei, the 3 nuclei form a 1 set of cell, they form antipodal cells. Another 3, a nuclei form 3 separate cell and form egg apparatus. These 2 nuclei remain at center. Hence, when you take a matured embryo sac, there is a presence of 3 haploid cells at chalazal end and 3 cells at micropylar end. And at center, 1 cell remain that we call central cell, and this central cell having 2 separate nuclei, they are called polar nuclei. Why do you call it as a polar nuclei? Because 1 nuclei came from polar nuclei. Hence, at the end of embryo sac development, there is a formation of 7 cells with 8 haploid nuclei. Then, if you take a structure of a matured embryo sac, which having a 3 haploid cells, they are called antipodal cells. And these antipodal cells always present towards chalazal end of the a megasporangium. The central cell, we also call it as a secondary nucleus, and it is present at the center of the embryo sac, it having two haploid polar nuclei. Whereas towards microphylar end, the large egg is there, that is ovum, which having a prominent vacuole with rich protoplasm, and later side two synergids are there. That is haploid egg and two haploid synergids together form egg apparatus. If you consider the synergids, their inner wall showing a cellular thickening, and that cellular thickening we call the name filiform apparatus. Filiform apparatus. That apparatus play a very important role to guide the pollen tube to enter into the directly egg apparatus or egg. Now we consider structure of megasporangium or ovule. Generally, ovule means it is future seed. Means after fertilization, ovule become seed. Now common ಆಗಿ ಈ ಪೊಮೊಗ್ರಾನೇಟ್ ಅಥವಾ ದಾಳಂಬ್ರೆ ಬೀಜ ನೋಡಿದ್ರೆ ಒಳಗಡೆ ಒಂದು ಸಣ್ಣ ಎಗ್ ಶೇಪ್ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಣಿಸ್ತದೆ ವೈಟ್, ಅದೇ ಓಯೂಲ್. ಅಥವಾ ಈ ಗ್ರೇಪ್ಸ್ ತಿನ್ನುವಾಗ ಒಳಗಡೆ ಒಂದು ವೈಟ್ ಎಗ್ ಶೇಪ್ ಸ್ಟ್ರಕ್ಚರ್ ಬರ್ತದೆ, ಅದು ಸಹಿತ ಏನದು ಓಯೂಲ್. ಇಫ್ ಓಯೂಲ್ ಫರ್ಟಿಲೈಸಸ್ ಇಟ್ ಬಿಕಮ್ seed. Basically, if you consider the megasporangium, it having a stalk that what we called the funicle. Stalk of ovule is we called funicle. Then inner margin of that funicle attached to ovule, that part is called hilum, and outer margin is called rape. Generally, if you consider each ovule covered by a coating called integuments. Actually, meaning of integument means skin it is. There are two integuments are there. One is outer integument. Later in seed, it become outer covering called testa. Second one, inner integument. It form later tegumen, the inner layer of the seed. And there is a presence of two ends. One is chalazal end and another one the microphylar end. Generally, these ends changes from one type of ovule to another type of ovule. At center of each ovule, there is a presence of nucellus. It is not nucleus, nucellus. It is a nourishing tissue that helps for nourishment of the embryo sac or female gametophyte. Then finally, if you consider female gametophyte, we called embryo sac. Each female gametophyte having 7 cells and 8 nuclei. Here towards chalazal end, there is a presence of a 3 haploid cells. They are known as a antipodal cells. Then we have central 2 nuclei. Their name is polar nuclei, and that cell is called central cell. Towards a microphylar end, generally there is a presence of large central cell egg, or that we also call ovum. It is rich in protoplasm, and later after fertilization, egg become zygote and develops into young one. Side we have 2 synergids. They are also haploid in nature, and these synergids generally having specialized piliform apparatus that guide pollen tube enter into the egg. In this part, especially one 5 mark question you are getting, that is development of female gametophyte or embryo sac along with diagram. One more a 5 marks question on only a structure of embryo sac or female gametophyte. The last question, sketch and label megasporangium or ovule and explain the structure. That means in this part we are getting 3 5 marks question. Really in this part, that is the term is still megasporangium and embryo sac. Little bit confusion is there. At least you have to make totally 5 units. In that first one, under introduction, just you have to give definition for pistil or gynoecium, then units called carpels, then ovary, style, stigma explanation. Whereas in megasporogenesis, only you have to develop megasporogenesis steps and also monosporic, bisporic, trisporic and tetrasporic condition. Whereas in third term, female gametophyte or embryo sac development, that diagram you have to prepare separately and explanation. Then structure of female gametophyte, that is a 7 cell and 8 nuclei diagram you have to draw and take the explanation. Last one, the structure of megasporangium or ovule you have to draw, and there is a little bit confusion. The microsporangium, that is a circular part found in anther, and megasporangium. These two terms are little bit confusing. ಅದಕ್ಕೆ ನಾವು ಇಲ್ಲಿ ಹೇಳುವಾಗ megasporogenesis, female gametophyte development, structure of female gametophyte ಮುಗಿದ ನಂತರನೇ megasporangium ಅಥವಾ ovule ಓದಬೇಕು. ಯಾಕಪ್ಪ ಅಂದ್ರೆ ನಮ್ಮ ಬುಕ್ಕಿನೊಳಗೆ ಫಸ್ಟ್ megasporangium ಕೊಟ್ಬಿಟ್ಟಾನೆ. ಅದಕ್ಕಿಂತಲೂ ಈಸಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ megasporogenesis, female gametophyte development, structure of female gametophyte ಪ್ರಿಪೇರ್ ಮಾಡಿದ್ದು ಅಂದ್ರೆ structure of megasporangium almost all 99% cover ಆಗುತ್ತೆ. Thank you.